All right, hello everybody. This week we're talking about the biology, what do you know, of photosystem 1 and photosystem 2 and the light reactions of photosynthesis. So we're going to get into this process in a little bit more detail. We're at this point where you kind of understand, hopefully, the overview of the process, sort of the ingredients and the products, where it happens, why it happens, why it's important, a little bit about light, that sort of thing. But now we have to kind of get into some details here, and the purpose of this is to get you guys to understand a complex process. So more than likely you won't need to know this in your future. However, definitely likely is the fact that you're going to need to be able to understand a complex process for whatever career that you decide to follow. So let's get into it, and the goal for you is to understand all the details and intricacies of the light reactions of photosynthesis. Here we go. The first key is to understand what ATP and ADP are. ATP is like chemical energy. Think of ATP as being like a granola bar or something like that. And if you eat it, it's going to create some energy. ADP is the energy molecule that has been used. It's like the wrapper of the granola bar. It's not really good for much of anything. Although we can use a battery analogy to compare ATP and ADP. ATP is the charged battery, and ADP, change that, is the discharged battery. So how do we go about charging this molecule which carries energy? Well, basically we add a phosphate group. If we understand that ATP is adenosine triphosphate, it's 3 phosphate groups, and ADP is adenosine diphosphate, phosphate with 2 phosphate groups, the chemical energy is stored in that 3rd phosphate bond. So that phosphate can sort of break apart and release the energy and become ADP, or it can be added back on, which takes energy and become ATP. So that's how our body, our muscles actually use energy, is through this process. So we use these same molecules that plants use. Another energy carrying molecule that we'll see in the light reactions is NADPH and NADP+. NADPH is the charged energy carrying molecule, and NADPH would be the uncharged version. So the difference between these two would be the hydrogen ion, as it's attached to the NADP+ molecule, takes energy to do that, and then when the hydrogen ion is released from the NADPH molecule, the energy is released. So the energy is in this phosphate bond, and the key idea here is we're going to look at an image here of the light reactions in a second. The ATP and NADPH is the goal of the light reactions, to essentially charge these energy carrying molecules, which takes energy, so that they can go into the stroma of the chloroplast and complete the Calvin cycle, which is where we take the carbon dioxide and we synthesize that raw carbon and turn it into glucose, C6H12O6. But that process requires a lot of energy. So we take the energy carrying molecules that we got from the light reactions, and then we use them to construct glucose. Then those energy carrying molecules, ATP and NADPH, will go back to the light reactions and sort of get recharged. So let's take a look at the light reactions here. Zoom in. Okay, so you're going to have to sort of know everything that happens in this diagram. And I'm going to sort of walk you through it step by step. The first thing to understand is that this is the thylakoid, and the thylakoid is inside of the stroma, and the stroma is in the organelle, where the photosynthesis takes place, the chloroplasts. And so this is a zoomed in view of the inside of a chloroplast. And if we remember from the leaf structure, most of the photosynthesis happens in the mesophyll cells and that spongy tissue inside the leaf. So the first question is, what are these things? What is this? There's a lot going on here. And so all of these things happen along the thylakoid membrane, this process. And so there's a lipid bilayer which creates this membrane, just like we learned about in our last unit. And outside is the stroma, sort of like this fluid, sort of like the cytoplasm. And what we do here is we have a bunch of proteins on the membrane which serve various roles in something called the electron transport chain. And ultimately the end goal is, like I said earlier, to produce NADPH and ATP. So let's go to this photosystem 2 to begin with. Photosystem 2 is a large protein embedded on the membrane that has a very specific role. What it does is it essentially absorbs light. So this is, light is important for the light reactions. And the light energy is used to split water. So here we have two water molecules which are being split apart into hydrogen ions and oxygen molecules. And notice how they're hydrogen ions. They're ions because they've been stripped of their electrons as well. And there's those 4 electrons that they lost for the 4 H pluses. And so this water, where did that come from? That came through veins in the leaf to deliver the water into the thylakoid space. And the water is important as a source of hydrogen ions. We're going to get to why those are important in a second here. But basically, photosystem 2 is the first photosystem that in this chain. It's called photosystem 2 because it was the second one to be discovered. But basically, in summary, photosystem 2 is the first step. It absorbs light, it splits water, and we produce free electrons. And we build hydrogen ions inside the thylakoid space. And you can see all these little blue dots in here. What we're doing is we're producing a bunch of hydrogen ions. And those are going to have an important role later down this through this process. So the electrons that were created here or liberated then move to the next step along the electron transport chain. And what they do is they power a protein pump to pump hydrogen ions from the stroma inside the thylakoid space against their concentration gradient. Typically, we would expect hydrogen ions that are all concentrated inside would want to diffuse out. So what we're doing is we're trying to pump more in and build up this concentration of hydrogen ions. And you can see where this source of hydrogen ions is. They come from sort of the end of this ATP synthase pump, which we'll get to in a moment. So basically at this point, we split some water, we liberated some electrons. Those high energy electrons sort of power this pump to increase the hydrogen concentration inside the thylakoid space. So then what we have to do is we have to re-energize those same electrons with more light through photosystem 1. And this time the electrons have a different purpose. Those electrons go and are combined with NADP plus to produce NADPH, along with 2 hydrogen ions found in the stroma. So it takes energy, like I said, to go from the uncharged NADP plus molecule to the charged version of the NADPH. So what we've done here is we've created a chemical which contains energy. But we also need ATP, and that's where the hydrogen ions come in. So this is the end of the electron transport chain. However, at the end of this process is something called ATP synthase. ATP synthase is another large protein embedded on the membrane, and its role is to charge ADP into ATP. How does it do that? It needs to add a phosphate group. And to do that, it's going to require energy. The energy doesn't come from free electrons in this case. The energy comes from the hydrogen ion concentration. Basically, it's like a water wheel where the water flows through the wheel and makes the pump turn. And that turning motion is energy, which can then attach the phosphate group to ADP, creating ATP. And so ultimately, the concentration gradient drives this pump to energize the ATP molecule. So at the end of the day here, we've produced 2 molecules of NADPH and 1 molecule of ATP as we move through this process one time. And I'm not going to hold you guys responsible for the numbers here, like 4, 2, 2, 4, 2. What I care about is that you understand where all these things go, like where the ingredients come from at each step, what's their purpose, and what are the products of each step. So let me go back through this again, because I bet you're confused. Generally, with a complex process like this, in anything in life, it's going to take a few times for you to really wrap your brain around what's happening here. So let's go back to the beginning. In the beginning, light energy is entering the chloroplast. It is absorbed by photosystem II, a protein embedded on the thylakoid membrane. The purpose of this light energy is to split water molecules, creating hydrogen ions and oxygen. And just as a side note, that's why we end up with oxygen as a product of photosynthesis. Really important, that first step for us. The hydrogen ions build inside the thylakoid space to create the concentration gradient, which drives the ATP synthase enzyme. Then, those liberated electrons move along something called the electron transport chain. The first time, what they do is they move hydrogen against the concentration gradient inside the thylakoid space. And you can see where that hydrogen came from over here. See, if we're constantly moving hydrogen ions out of the ATP synthase, we have to pump them back in too, otherwise we'll run out of hydrogen ions. Then those electrons are re-energized. So they use their energy first to move hydrogen ions inside. Then they use their energy again to construct the NADPH molecule. Finally, the ATP synthase pump works on the basis of hydrogen ion concentration gradient attaching a phosphate group and generating an ATP molecule. And what is the purpose of these two energy-carrying molecules? Well, they go on to the Calvin cycle, which happens in the stroma of the chloroplast, which is this area out here, and we start constructing sugar, glucose, from the carbon dioxide molecule. And if you care, here's a picture of the Calvin cycle. And we looked at this a little bit with our simulation last week. I'm not going to hold you guys responsible for this. I think one of these processes in detail is probably enough to give you some practice to really have a deep understanding of a complex process. And if you're still confused, watch this video again. And of course, I'll help you guys.